Okay, so we were discussing cleavage in the last class. We finished, I think, cleavage of Caenorhabditis. So today we'll discuss the cleavage of Drosophila, of amphibians, basically frog, and then fishes and birds. Okay, cleavage in all these animals. So all the, you can say, the questions of part C you can expect from these topics. Sorry, part B you can expect from this particular topic. So what all things you know about Drosophila? Have you seen Drosophila? Huh? Everybody? Yeah? Good. Because when I was studying Drosophila during my student time, I never understood what is Drosophila because I thought that I have never seen Drosophila. So it was just a big kind of animal, and I'm from zoology background, so you can understand if you have not seen the animal, it's very difficult to understand. Okay, so I just used to imagine, okay, some or the other kind of animal it will be. But later I got to know, okay, the all the different kinds of flies, small flies, they are nothing but some or the other species of Drosophila. Okay, fine. So today we'll start with the cleavage in Drosophila. I'll just fine. So cleavage in Drosophila, it's of superficial type. That's why I've taken this example. It's very important to understand because each type of cleavage, as in amphibians or fishes or birds, each type of cleavage is very important, and since we will discuss it in detail, so you will also understand what all things are related with superficial cleavage. Just we know that this cleavage is something which is superficial, which is not proper. That's why it is called as superficial cleavage. So in this cleavage, first what we see, we just see the division of nuclei. Okay, so if and once the cell has got so many nuclei, after that these nuclei, what they do, they move towards the membrane area, and there they undergo cellularization, or you can say cells are formed once the nuclei reach at the nuclear membrane, or not nuclear membrane, the plasma membrane or the cell membrane of the egg, right? Okay, so all these things take place step by step, so we have to follow that particular step. Okay, so after how many steps or after how many nuclei these nuclei will migrate, or after you can say fifth cleavage they will migrate, or after seventh cleavage these nuclei will migrate. We have to know all these things as well, right? Okay, so fine. Now this is a big kind of a chart. I'll just close this one. Fine. Yeah. So it starts with something. First, in this chart you have something in big boxes in which nuclei, the number of nuclei is written, and something in blue blue circle, and in that 1, 2, 3, 4 kind of it is written, and that is nothing but the cleavage. So after first cleavage, one nuclei inside the zygote, whenever an egg undergoes fertilization, it will form one zygotic nucleus. So one nuclei will undergo division, it will form two nuclei. So after first cleavage you will get 2 nuclei, second cleavage you will get 4 nuclei, third cleavage you will get 8 nuclei, fourth cleavage 16, fifth cleavage 32, sixth cleavage 64, seventh cleavage 128, and after 8th cleavage you will get 256 nuclei. Okay, now after 8th cleavage, actually the migration of nuclei towards the cell membrane starts. Okay, migration of nuclei. Once the total number of nuclei in the embryo is 256, then these nuclei, they try to migrate towards the cellular membrane. Okay, so after 256, 256 is the number after 8th cleavage. So after 9th cleavage, when there will be 512 nuclei. Okay, so out of you can say 5 nuclei, out of 512 nuclei, what they will do, they will migrate first. Okay, they will migrate first to the cell membrane. Till now these nuclei, they are dividing in the cytoplasm. They are nothing but they are present in the cytoplasm. Okay, it's just like this. Already we have seen the in earlier picture itself. It's just like this. So nuclei is dividing in the cytoplasm, right? So nuclei will keep on dividing. Once the total number of nuclei is 512, then 5 nuclei out of 512 they will start migrating. Okay, and they will migrate to any area, and that area will form posterior pole. Are you able to understand this thing? Okay, yeah.
After 8th cleavage, they migrate towards.
Not after 8th, after 9th cleavage. After 9th cleavage, once 512 nuclei is formed.
What happens after 8th cleavage?
After 8th cleavage, 256 nuclei is formed.
Okay,
After 9th cleavage, 512 nuclei is formed. Okay, so each nuclei is undergoing a division. And this cleavage or something is symmetric. All the nuclei is undergoing division at the same time. So it's not asynchronized kind of division. It's synchronized. So all the nuclei are undergoing division. So after till 8th cleavage, there is no migration. All the nuclei will be present in the cytoplasm only. But when 9th cleavage will take place and 512 nuclei will be formed inside the cell, then 5 nuclei out of 512 nuclei will migrate towards cell membrane. Okay, fine. Now why only these 5 nuclei will migrate? There are some mRNA, there are some proteins which will be associated with this nuclei, which will guide the nuclei. Okay, in zygote we know that what kind of what all proteins are there. The maternal proteins and maternal mRNA are there. Those mRNA and those proteins will guide these nuclei to migrate to some area, and that area will be developed into what? Posterior pole or posterior side of the embryo. Understood? So those 5 cells, wherever they are migrating, that area will form the posterior side. Once they migrate, okay, they will undergo cellularization. They will form cells there. So 5 nuclei will form cells, and other nuclei, 507 nuclei will not form any kind of a cell. 507 nuclei will remain in the cytoplasm. Only 5 nuclei they have moved, and they have, after moving, what they have done? They have formed a cell there. Okay, at the posterior pole. Now what happens to the 507 nuclei? They undergo second division, and that is called the 10th cleavage. Okay, cleavage with the bombardment. Fine. Okay. So what I am saying, that after the 5 nuclei, they migrate to which area? They migrate to the poles, or they migrate to some area that will form the posterior pole. The 507 nuclei which are there in the cytoplasm, they will undergo what? They will undergo 10th cleavage, and they will form some nuclei, right? After they form some nuclei, 507 nuclei will undergo division. How many nuclei will be formed? 1014. 1014 nuclei will be formed. 1014. Only 507, they are only undergoing division. So 507 into 2, that is 1014. Okay. So those 1014 nuclei will now migrate towards the membrane. You understood? So in 9th cleavage, after 9th cleavage, only 5 nuclei migrate. After 10th cleavage, 1014 nuclei migrate to the cell membrane. It's clear? Okay? To everybody? Follow me is also there. Follow me, you also can respond in chat. Okay, it's clear to everybody? Now, once the 1014 nuclei, they have migrated towards where? They have migrated towards the cell membrane. That will form a kind of a structure, a kind of an embryo, which has got too many nuclei and no cell, or only 5 or 6 cells. Because till now only 5 cells are formed, and other nuclei, they are just as it is. Okay, and that particular embryo is called as syncytial blastoderm. Now, why it is called as syncytial? What is called as syncytial? I'll show the diagram.
Less of cytoplasm.
This is what is your syncytial blastoderm. Okay, now syncytial blastoderm. Now here what we have? We have, these are nothing but what kind of cells? They are the polar cells. Okay, they are the polar cells, and these are the nuclei which migrated the 1014 nucleus. It was impossible to show 1014 nuclei, so I have say some 15 or 20 nuclei. So these nuclei, oh, one second. So these nuclei, they are also nuclei, and they have migrated towards this area after 10th cleavage. So after 10th cleavage, what you get? You get a syncytial blastoderm. Syncytial blastoderm. Now, why the name is syncytial blastoderm? What is blastoderm? Okay, tell me, what do you understand by ectoderm? Outermost layer, which forms the outermost layer. Okay, germ layer. It's a kind of outermost germ layer. So blastoderm, germ layer, blasto. Derived from the blastula. Yes, derived from the blastula stage or derived from blastomeres. You understood? Because when cleavage will undergo, what you get? You will just get blastomeres. You will get what? You will get a blastula. You will get a blastula. Okay? And in this case, what you are saying? You are seeing a layer of cells that will be formed later, not right now. You are seeing what? Layer of nucleus, which is present near to the cell membrane. This is nothing but the cell membrane. Okay, this is cell membrane. I'm just writing MEMB. Okay, this is nothing but your cell membrane. The nuclei, they have migrated towards the cell membrane, and what they have formed? They have formed a germ, they have formed a layer. So derm, okay, and blasto, why? Because this will actually lead to what? Blastula. After cleavage, you will get a blastula. So yes, this is the blastula stage. That's why it is blastoderm. Why it is syncytial? Syncytium, fungi. In case of fungi, multinucleated cells are called as syncytium. So this is a multinucleated cell or not? Sorry? Each of the nucleus, yes. Each of the nucleus is not there. Also, all the nuclei is present in the same cytoplasm. So if the nuclei is present in the same cytoplasm and there is no demarcation of the cytoplasm, then the cell is called as a syncytium. Tium. Okay. So why it is called as syncytial blastoderm? It's clear to everybody, online as well as offline students. Fine. So till 8th cleavage there was no problem, but when 9th cleavage happened, the cells, the polar cells were formed. Okay, and when 10th cleavage happened, the number of nuclei increased and they migrated towards our cell membrane. Okay, now what is the concept of polar nuclei? Polar nuclei or polar cells, not polar nuclei, polar cells are those cells which will be responsible for the formation of gametes, reproductive cells. They will form the reproductive cells. That's why they are present at the posterior pole, and they will form the posterior pole, so the other side will be developed into an anterior pole. It's as simple as that. So why they are called as polar cells? Because polar cells are generally responsible for the production of gametes, right? Okay. So this is syncytial blastoderm. Now after this also cell division will take place. Sorry, nuclear division will take place. So this is a stage after cleavage 10 till cleavage 13. This will be the stage. Till cleavage number not 13, till cleavage number 12. This will be the stage. So cleavage number 12 means what? If cleavage number 11 I am saying, that means each nuclei which is present near the cell membrane, there only it will undergo division. There only it will undergo division. So each nuclei, for example, if I talk about this particular nuclei, okay, so if this nuclei is present over there, so this nuclei will form two nuclei at that area only. Then this nuclei will further form two more nuclei. This nuclei will further form two more nuclei. Each nuclei will keep on forming two more nuclei. So there will be, you can say, overcrowding, overcrowded area at the cell membrane. Until now, no cellularization has taken place. Cellularization will take place after cleavage number 13. Okay, it's clear to everybody? Follow me, it is clear? Fine? Yeah. Yes, yes, till the 12th cleavage only nuclear division will take place. When you will see 13th cleavage, you will say cells will be there. So 14th cleavage is a cell division, karyokinesis followed by cytokinesis. Okay. After 13th cleavage, you will get cells, that is for sure. So 11th and 12th cleavage, that is, you will just get what? You will just get the nuclei there. Okay. Are you following this? So now one more thing is, now these nuclei, they are called as energids. Not only this nuclei, okay, don't get a misconception. Not only this nuclei, the nuclei along with the cytoplasm which is surrounded by, which is surrounding this particular nuclei. Okay, so the nuclei, this is the nuclei, you just color it, okay, and the area of the cytoplasm which is surrounding this particular nuclei is called as energid. So what is energid? Energid is equal to nuclei plus cytoplasm, local cytoplasm, you can say, local cytoplasm, and that local cytoplasm is called as cytoplasmic island. Okay, cytoplasmic island. Okay, so what is energids? Energid is not the nuclear plus the local cytoplasm. Now in that local cytoplasm, what is present and why that is different from the other cytoplasm? Okay, in that particular area, you will see lot of microtubules and microfilaments in that area. Why microtubules? Because karyokinesis will take place. Why microfilaments? Cell membrane is near. No, cell membrane. Cytokinesis will take place, but not right now. It has to take place. Okay, cell membrane. Near to the cell membrane, always microfilaments are seen. Okay, and they are actually responsible for taking the cell membrane in and blowing the cell membrane out and all those things. Okay, so microfilaments are involved in this one. So microfilaments and microtubules will be seen in that area. That will form a cytoplasmic island across this particular nuclei, and since it has not got its own cell membrane, that's why it's not called as a cell right now. It's just a energid. So this energid will, yeah. Microfilaments will help in cellularization, in the formation of cell membrane across this particular nuclei, because they will, what they will do? They will pull the cell membrane. Okay, then only the membrane will follow this particular nuclei and you will get a cell. I will tell you that also. Okay, fine. Now, so that was just the syncytial blastoderm. Now after syncytial blastoderm formation, that means this particular structure formation in which 5 cells are there, plus one big cell which contains 1014 nuclei. This is what is syncytial blastoderm. It is followed by, yes, follow me, you are asking something? Every million cells of blastula will perform karyokinesis together. Yes, it's not million of cells, it's 1014 cells. Sorry, nuclei. Each nuclei will undergo karyokinesis together. Yes, there is synchronization because in mid-blastular transition only you will see asynchronization. Okay, so till now there is no mid-blastular transition. It's still in the which stage? Before blastula. It's before blastula. Okay, fine. So after the 11th cleavage, what you get? You get mid-blastular transition. Okay. So after syncytial blastoderm, the cell enter mid-blastular transition. Now in mid-blastular transition, what all things happen? Transcription of genes will take place. Some proteins will be formed. Asynchronization of the nuclear division or the cell division will be seen. All these things. Now, in case of Drosophila specifically, what is seen, what is observed is nuclear division will slow down. Nuclear division. Sorry? After the 11th. After the 11th cleavage. So, yes, once the 11th cleavage is over. Now, 11th cleavage is taking place where? Near to the cell membrane. Nuclei is present near, na? So there only cleavage is taking place. Okay, so the nuclear division will slow down. Okay, earlier the duration of nuclear division till 12th cleavage, not 12th cleavage, till 10th cleavage, or till 11th, not 10, you can say till 11th cleavage, the duration was 8 minutes. After every 8-minute nuclei was dividing, each and every nuclei in this particular cell. Okay, in this particular syncytial blastoderm. Fine. And in mid-blastular transition, after, you can say, or in the mid-blastular transition, the time duration is from 25 minutes to 175 minutes. You can imagine the duration increase. 25 to 175 minutes it is taking now. Okay, so in Drosophila it is 25 minutes, in some other species it is 175 minutes also. So it is taking some time, and in that time what will happen? Nuclear gene will undergo transcription, some proteins will be produced, translation will follow, all these things, and also microfilaments will start pulling the cell membrane, all these things will follow. Okay, and rate of transcription increases. That is what I already told you. That is generally seen in mid-blastular transition. Now, after mid-blastular transition, in cleavage number 12, normally the nuclei will keep on dividing. Okay, nuclei will divide. Fine. Okay, so till 12th cleavage, nuclei will normally keep on dividing. So 11th and 12th cleavage generally marks the mid-blastular transition phase. So in 11th and 12th cleavage, slowing down of the nuclear division, your formation of some proteins, transcription of the genes, all these things will take place. Now when the 13th cleavage is there, after 13th cleavage, what you get? You get a cellular blastoderm. Now that is a syncytial blastoderm. It will be converted into a cellular blastoderm. Okay. So syncytial blastoderm is there, and that syncytial blastoderm has got only nuclei. Now cellular blastoderm will have only cells. Simple to understand also, and why it is blastoderm? Because this is in cleavage, blastula stage, and this is just forming a monolayer, so that's why it is a blastoderm. Okay, fine. So you just please make sure that you understood the structure of syncytial blastoderm, because when I'll click, we will get what? We will get, oh, we are not getting anything. Achha. So yeah, this is what is the cellular blastoderm. Now, in cellular blastoderm, what all things are there? Now each cell, actually it was very difficult to put nuclei to each cell, that's why I didn't put nuclei there. I thought okay, I'll put that in class. So each cell has got its own nuclei. Okay, fine. Now you are seeing that okay, these are the cells formed formed after compartmentalization. Already nuclei was there, just the invagination of the plasma membrane led in the formation of the cells. So cells are formed, right? Okay. And cells are formed and cell will contain what? Nuclei it will contain. And there is a mid part, this part. What is this part? This is the yolky part. Yolk. So now yolk has also got partitioned off from the normal cells. So cell has just formed a monolayer around the yolk. That was what we saw in the earlier picture or earlier diagram also. In superficial cleavage, that monolayer of cells will be formed. So in the 11th cleavage, 12th cleavage, the invagination of the plasma membrane will keep on happening, and ultimately at the end of 13th cleavage you will get a cell like this. So that is what is mid-vascular transition. Okay, fine. Yeah. What? Cellular blastoderm. Cellular blastoderm is produced from the syncytial blastoderm, that is there, but it happened after 13th cleavage. So when 13th cleavage happened, you got some cells, okay, in spite of getting only nuclei, got some cells, and these cells will form a monolayer around the central yolky part. These cells, they have formed, the green colored boxes are nothing but these single cells. They have just formed a monolayer around the central yolky part, and each cell has got its own nuclei. Okay, so each cell has got its own nuclei. So what actually has happened, since for example this is the plasma membrane and nuclei is there, okay, so plasma membrane actually did what? It tried to fold like this. So nuclei is present here and it just tried to fold, and it kept on doing that, and ultimately it fused with the lower layer to form the cell membrane. Those cells, the specific name is blastomere. Okay, apart from that there is no specific name. Fine. Okay, so single or the blastodermal cells also you can say, because they are forming a dome or they are forming a layer. Sorry? This is not completely blastula, right? There is no empty space, that's why.
Yeah, but then why we say blastoderm?
Okay, see, whatever is happening in cleavage, that will lead to blastomere. That will lead to blastomere formation. And wherever blastomeres are there, blastomeres are forming a monolayer, and that will be called as a blastoderm. Okay, so this is something blastula. So each and every animal will have a different type of blastula structure. So we cannot say that it has to have a central big cavity. Here also this big cavity is filled with yolky material. The cavity is there, but the cavity is filled. There also cavity was there. Cavity was there also filled with the proteinaceous material. Here it is filled with the yolky material.
Cellular blastoderm can be called.
Yes, this is what is blastula. Yeah, this is what is blastula. Fine. Okay, any other question you have? No. So this was just the overall picture. Now we have to go into little bit detail, and that is what kind of detail? How these cells are formed. So from normal nuclei, how this plasma membrane invaginated, it's very simple to understand also. There is the invagination of plasma membrane with the help of microtubules and microfilaments. So when we will look at the diagram of the picture, you will understand. It's very easy to understand also. Okay, so till now it's clear to everybody? Fine.